We are also live streaming today on YouTube, so anybody who wants to join us using the YouTube format, you're welcome to join us over there as well. We're going to get started in just a couple minutes here. Hello from Cambridge. From CRLS High School, excited to learn about 3D cell images. All right. So we're going to just give a few more minutes here. Another minute. Hello from Shoreline. Hello, neighbor. And we're excited to hear about cells. Great. Excited to learn about the technological side of biology from Bellingham. You're in the right place for that. Great. Well, we're going to go ahead and get started. So this is Behind the Scenes of Science. My name is Caitlin, and we are here today with Mackenzie Coston, who is a research associate in the Allen Institute for Cell Science. So the goal of today's webinar and the whole Behind the Scenes of Science series is to give you a sense of some of the careers in science, different pathways, and how you go from high school to now. What does a day in your life look like? So I'm going to ask Mackenzie a few questions to get us started, and then we'll be able to take some questions from you in our audience. So if you look down at the bottom of your screen on Zoom, you should see a button that says Q&A. So if you want to go ahead and use that button to enter any questions that you have for Mackenzie, I'll take those later in the session. And then if you're watching us on YouTube, we'll also take questions from YouTube. Just go ahead and use that chat box. So I'm seeing more introductions coming in. While Mackenzie talks, please feel free to keep introducing yourselves. So Mackenzie, to start, what was your science journey from high school to now?
Yeah, so hi everybody. I actually grew up in Bellingham, so exciting to see all the folks from Bellingham. And I went to Squalicum High School as well. I took biology with Miss Loss my freshman year, went on to chemistry my sophomore year. Loved the science, always thought it was super interesting. And then I did running start as a junior and a senior. So I was at Whatcom Community College, able to take more chemistry, more biology, and kind of take my science to another level, which was really fun. I then went on to the University of Washington in Seattle. It's kind of always where I wanted to go. They had a great science program, so it aligned with what I wanted to do. As soon as I got to UW, I started looking for research opportunities, emailing anyone and everyone who looked interesting to me. I finally got some interest from a cancer genetics lab, which was really neat. So that was my first foray into research. I was basically just doing data entry, which was not the most exciting, but got my foot in the door, which was really important when getting into science. I then went on to do a summer program at UC San Diego through the National Science Foundation, which was really amazing, in cancer cell mechanobiology. That was super fun. Came back to Seattle to continue with school. And then I got a job at Harborview Medical Center in the neuropathology group, which was totally different than anything I had done before. I got to work with brain tissue and do all sorts of cool things like flow cytometry and work with antibodies and staining, which was really neat. I then did an internship at the Infectious Disease Research Institute. So another change of what I was looking into and studying, where I looked at tuberculosis and bacteria and drugs. And then I went on to study asymmetric cell division during my senior year at UW, which was really neat. We used fruit flies for that research. So another totally different field of research, essentially. And in that research, I did a lot of microscopy and 3D imaging, which was really neat. I'm a visual learner, so for me, microscopy is really cool because I get to see everything. And after I went through UW, I didn't really know. I initially wanted to do bioengineering, But as I continued studying science a little more and doing research, I realized that cells are awesome, and I would much rather just spend my time studying the cells and how they work. So that's what led me to molecular, cellular, and developmental biology, which is my degree. And then as I was finishing my senior year, I found this job at the Allen Institute, applied, went through the interview process, which is a fun time. And luckily, all of my studies and prior research experience really prepared me well for this job, and I got it. And I've been working here for about a year and a half now, which is really exciting.
Well, that's a great segue. Can you tell us about what does a day in your work look like, both before the pandemic and now?
Yeah, so prior to the pandemic, since I work on the microscopy team, we do a lot of imaging. So on a typical imaging day, I would get in pretty early and we image cells, specifically stem cells that we have genetically edited in a way where we can see certain structures within the cell light up with fluorescence, which is pretty cool.
Before we go on, can you tell us a little bit more about what those cells are and where they came from?
Yes, so they're called HIPSCs, or human-induced pluripotent stem cells, which is a mouthful. But they are cells that have come from one man's skin cells, which is kind of cool. And then we turn them back into stem cells. And stem cells are kind of a primitive type of cell that can become many different types of cells. So they're really great when you're studying cells because you can translate the research from the stem cell into other types of cells, which is neat. And we edit them using CRISPR technology, which you may have heard of, kind of a buzzword. But essentially, it's a molecular scissors where we can go in, cut part of the genome out, and insert genes we want. And when we insert the genes, we are tagging them with fluorescence. So we put green fluorescence protein right in front of other proteins that are part of the mitochondria or microtubules, different parts of the cell that we want to visualize. So then when we go to image these cells and we hit them with fluorescence in the microscope, these things light up, which is super cool to see. So in order to image these cells, you have to grow them and passage them, which is part of my job. So when you grow cells, you put them on a surface or a plate or a dish, and you let them grow for about 3 to 4 days where they reach a nice confluency. And then you pick up a subset of these cells and put them into a new dish. So that's called a passage. Every 4 days, you take new cells, put them in a new dish, and so on. Most people do this by hand, but at the Allen Institute on the microscopy team, we do it using a robot, which is pretty cool. So that's a large part of my job as well, making sure the robot is functioning, make sure nothing's gone terribly wrong, and then looking at these cells, making sure they look healthy, making sure we have enough of them for the day's imaging. We have to prepare certain dyes for the cells the morning of. Turning on the microscopes is a whole process. These are really kind of complicated microscopes. It's not just the simple little ones you may have used before in high school. These are 3D live cell imaging microscopes. So we can see in the X, Y, and Z plane, which is really neat. And then we use a blue fluorescence to look at the nucleus. We use the green fluorescence to look at whatever labeled structure we want. And then red fluorescence to look at the plasma membrane. So imaging generally takes all day. So that would be an all-day event of making sure you've set up the plate correctly and focused on the right areas of the cell, and it's collecting how you want it to collect. But some days it's also meetings all day. That can be a common thing in science and research, lots of meetings, data analysis especially. And now during COVID times, it's a lot more data analysis because that's very easy to do at home. You don't need cells and you don't need pipettes and things. So my daily work has definitely pivoted with COVID. The first few months of quarantine, I was just solely working at home on data and helping to write a paper about the robot in our automated cell culture system, which was really interesting. So making figures, making graphs, a lot of creativity in that too, which you might not expect, but pretty fun and different. And then I was able to start going back into the lab in the end of May. So I went in once every 4 days to passage the cells. So a good balance of still going into the lab and seeing the cells, but also doing some neat data analysis at home.
So that cell passaging uses the robot. Would you like to talk about how that works while I show the video?
Yeah, so we have a really cool video. This robot, it's a Hamilton robot, so we just call it Hamilton because every robot needs a name. So this is the robot. It's pretty cool. It handles plates and lids and troughs, and it is connected to a refrigerator and an incubator and a centrifuge. If you know what that is, it essentially spins things really fast. So we have the ability to do a lot of stuff automated, which is really awesome, because normally in cell culture work, in cell work, cells grow in kind of a media broth. They grow in a liquid, and this liquid has to be changed every day, which means someone has to be working every day, generally on the weekends. But with the robot, we don't need that, which is really nice. So it can be changing the media at 5 in the morning, and then you come in and your plates are ready to go. So that's really cool. Here you can see like the pipette tips going across the plate. It's cool too because this is a really customizable system. So you work with the company and tell them what you need, and then they can adjust. And it's definitely requires a lot of creativity to adapt the manual protocol to a robot. You have to do things in completely different, weird ways. Because when you're pipetting with your hands, you're able to hit different angles that a robot can't when it's just kind of moving in a linear fashion. So my partner Ben and I have gotten very creative.
So we have a question from somebody, did we make the robot?
So yes and no. So we worked really closely with Hamilton to design the robot. We actually are in the process of acquiring another one as well. And I've been able to be in some of the meetings with our Hamilton representative, where we all sit together and brainstorm, okay, what would be helpful to help us do this protocol, and what type of things should we add on? So on our next system, for example, we're going to have an articulating arm, which is really cool, which is kind of what people think of when they think of like a robot. So in a way, the sky's the limit. If you can think of it, they can help you build it. But we did not make it specifically. That was the Hamilton folks.
Great. Well, our next question is, what's your favorite thing about your job? And I think you've covered this a little bit just now.
Yeah, so I mean, working with the robot is really interesting. I love the creativity aspect. I'm a creative and a visual person. So like I was saying, the microscopy is really awesome for me too, because you put these cells on the microscope, you turn the scope on, and you see the inside of a cell, which is so cool. That never gets old. Especially sometimes time-lapse images or videos, so pictures of cells over time where you can see them dividing on such a magnified level. It's pretty cool.
I've also pulled up an image of sample cell to show. So these are the cells that Mackenzie is talking about. Here's one.
Yeah, so this is a typical, well, no cell is truly typical, but as you might see by going through our cell library, but it definitely looks different than what most people think of as a cell. I mean, it's not, they're hardly ever perfectly round, and there's so many different dynamics going on. So here you can kind of see what I was talking about with the green fluorescence and the red fluorescence and the blue. So that red is the plasma membrane, and then let's see, this is Sec61. So that's what's labeled with the green, and then the blue would be the nucleus. And we have approximately 30 plus cell lines with different things tagged. So
our public image library currently contains about 32,000 cells.
Yes, so if you're ever curious about what a cell, the diversity of cell images, there's a lot of diversity here. And yeah, so they all look different and a little funky, right? And one cool thing about these cells is that they grow in a colony. So they're not just one cell sitting alone by itself. It's tons of cells packed in tight. So that's why they kind of almost look squished or have some interesting dynamics, and they're not just circular.
Yeah. So this is going to be, I think, another robot-related question. What's the funniest thing you've ever seen in lab?
So the robot produces a lot of both funny and frustrating moments. The robot is not perfect. You can program your movements down to the micrometer. You can tell it how to move down to the T, but still, if something isn't in the exact right spot, it can pick something up weird and things can go awry. So an example of this is one step in passaging is you put the cells into the dish called seeding. So you seed your cells onto a dish. And normally, if you do this manually, you shimmy your dish around a little bit to spread out the cells to make sure they're evenly distributed. And to do this on the robot, we have these two grippers that pinch the side of the dish. And then it picks it up and it zooms it around the hood. So inside the robot, it picks them up and it zooms this dish around the hood. And my desk is located outside of the lab, but we have big windows so I can look in and make sure things aren't going badly. But I peeked in at this step and I noticed it was just zooming around the lid. So the dish was not there, which was very concerning. So I went in and the robot had thrown, it must have picked up the dish off balance. And as it was zooming it threw the plate of cells out of the robot into the room. So I just saw a plate laying on the floor with liquid everywhere. So the cells were just all across the room. It's like, oh no. So there's all sorts of mishaps with that. But you have to laugh at it. And luckily cells, they do grow back and we have lots of them. They're always replaceable. But in that moment, it can be hard after you've spent the whole day passaging and then your plate has been thrown out of the robot.
It's good that you can bounce back from it, both scientifically and also just in the course of your workday.
Yeah, that's a big part of science. Things tend to not go to plan. So you just got to keep trying.
Yeah. Well, we have some questions from our audience. What are you trying to accomplish or what are you trying to find with your research?
Yeah, so our research is unique in that it's basic science. So it's kind of a broad thing. We just want to study cells and the dynamics of cells and to figure out how cells actually work and look. We have the 3D mitotic cell viewer, which is a comprehensive 3D look at all the structures within the cell. And then we are also really focused on making sure all of our data is publicly accessible and that we're creating tools regarding cells that everyone can use. So we want to figure out all the basics of cells and tell everybody about it so that they can use that information to further their own research. So it's a basic science thing in that we just really want to figure out, okay, how do cells work? What are the dynamics of cells? But also, how can we produce data and research that can further everyone else's scientific goals?
Yeah, that's a great comment that you made also about what basic science is. So just to elaborate on that a little bit, basic science is interested in like the fundamental properties of the world around us. In this case, cells, but it can be any field of science. And translational research is things like developing drugs or treatments for diseases. So we're really interested in basic science at the Allen Institute. Another question, how do you take out microscopic parts of the genome? So I think this is a question about CRISPR.
Yeah, so we have a stem cells and gene editing team, which they're the experts in this. I don't work over there. But essentially, CRISPR is an enzymatic protein, molecular scissors, as people like to refer to it. And you can make it so that it finds certain regions of the genome, so it's specific. and then it cuts with enzymatic activity, and that part comes out, and then it inserts your gene of choice. So that's a very high level overview. I'm not the expert in that, but I would definitely, we have a lot of resources that explain that on our website too, allencell.org. The gene editing team is very knowledgeable, and they do a lot of cool work with that.
Yeah. So we have a question from the YouTube viewers. So you have to deal with a lot of different specialties: data analysis, data visualization, cell growth, robotics, etc. Do you deal with imposter syndrome, and how do you manage having to know so many different specialties?
That's a great question. So especially for me, most of my colleagues are PhD level or higher, and I have a bachelor's. I am newly working at the Allen Institute. So coming in, it was kind of a shock because these people are all experts. They're amazing. They know so much, and it's hard to realize that I know a lot too, and I'm here for a reason. They didn't just hire me to hire me. They hired me because they thought I would be able to work hard and fit in well. So I think recognizing that is really important, and also making strong connections with the coworkers. I mean, they're just people too. They're just scientists. They know a lot, and then taking advantage of that too, asking them questions, especially when I am trying to do so many things. Going to the people I work with who are experts in those separate spheres of robotics or data analytics, not being afraid to just go to them, ask questions, learn as much as I can, and then always be accepting that I'm not going to know everything, but there's always a lot to learn, and people are always willing to help you learn.
That's one of the most exciting things for me about science, is knowing that you'll never know it all.
Oh yeah.
There's always something new to learn.
Yeah.
So actually, on that note, we have a related question. Is there always something to do? Is there ever a slow day? And how do you find new things to study?
Yeah. So there are slow days. Almost less so now working at home, which is interesting. But I think that's because there's a never-ending stream of data that needs to be analyzed. When you're in the lab doing experimental work, there are definitely slow days, or experiments that have long incubation times, or even imaging. It can take 2 to 4 hours of just kind of, you sit and wait for your experiment to be done. So definitely slow days, but always data to catch up on, papers to read. There's always something to do, but it's not always super cool, fast-paced, exciting robot doing cool things. And then, sorry, what was the second part of that question?
How do you find new things to study or learn?
Yeah, so that is a great question, especially since the Allen Institute is basic science. So we have a lot of freedom in what we choose to study. So that means we have a lot of leadership that decides what we want to study at a high level, what we think is feasible to study, what we think is needed to study in the field, and what research would be most beneficial for everybody to benefit from. So we have a lot of experts who come together and brainstorm and think. Since I'm a research associate, I'm more of a doer, not a high-level studier or planner. But the neat thing about this, if I had a little side thing that I was really interested in, I could totally bring that up with my team, and we could talk about it, and we would have the ability to explore without constraint, which is really neat about the Allen Institute.
What do you wish you had known when you were a high schooler about a career in science?
It's a very windy road. So like I had said, I wanted to do bioengineering, and I kind of thought I knew exactly what I wanted to do, but things happen, and I found that it's really important to just take opportunities as they come up, especially with science. If there's a research opportunity that appears to you, and it might not be your first choice or what you initially wanted to research, I'd say go for it. It never hurts to try something different or new and have that experience under your belt. And doing any type of research or work in science, I think is going to benefit you as a scientist in the whole. So it's not a straight path and it's very windy, but all of those twists and turns make you a better scientist.
Do you have any hopes for where those twists and turns might lead you in the future?
Grad school. But I'm still not entirely sure of what I want to do for grad school. So I'm still taking it one day at a time. I'm still twisting and turning. My path is not straightened yet, but that's fun and exciting. And I've come to embrace that and enjoy new experiences and challenges.
Yeah. So we are getting towards the end of our time. We may not get to all of your questions, but I'm going to get through a few more at least. How do you publish your results? Since you're looking at a very broad subject, will you publish all of your findings at the same time?
So no, we publish in a lot of different ways. Normally people think of publishing as kind of writing an article in a scientific journal and it's published there, which we do. For example, right now I'm writing the paper about our automated robotic cell culture methods. So that's going to be kind of a methods paper of this is how we did this. This is all of our methods and our code. So people can access that and hopefully replicate it for themselves. But we also self-publish a lot. So a lot of our resources like the integrated mitotic stem cell and allencell.org, we release our data in a lot of different ways. So we have software that's available for people. We have literal data that's available for people. You can get our cells, you can, at the Brain Institute, you can get mice. So it's a lot of really interesting releasing. So it's definitely bits and pieces released in a lot of different ways.
Yeah, so we release all of the data. The Allen Institute practices an open science approach. All of the data gets released, so you can go check it out on our website. And you can use it for your own research or your project in your class. If you're doing a science fair project or independent research as part of a class, you can use our data. Just allencell.org. I just dropped the link into the chat. Another question from the audience: is it difficult to have time for hobbies in this line of work?
Yes and no. I prioritize my work-life balance for sure. So I make time for hobbies. When I was pre-COVID going into the lab, those are definitely can be long-ish days. And sometimes it feels like you don't have any time for hobbies. But I think that can be any 9-to-5 job. But that's kind of the beauty of being remotely working now and working from home. You do have a little more flexibility. You're not traveling to work, which can take a lot of time, especially if you're in a big city. But yes, I prioritize my hobbies. I like to be outside for sure. I like to hike and go on walks and runs. I love to read, baking, which is most scientists love to bake. So it's like science with food. And then I also crochet, which is a fun creative thing for me to do.
I think we have, let's see, time for maybe one more question. We have a cell bio question. Do you use viruses to introduce CRISPR and proteins of interest into the cells?
Yeah, the gene editing team does use viruses to edit. I don't know much of the specifics, but they do. Again, I think more of that info would be on the gene editing portion of our website.
All right. Well, I think we are just about out of time. We have about 90 seconds left. Do you have any final thoughts that you want to share with our audience today?
Oh man, science is fun. You should always be willing to take these new opportunities in science if you're interested in it. Keep an open mind, be willing to learn. It's very rewarding. Yeah, stick to it. Don't get discouraged.
That's a great parting thought. So we hope we'll see you at future Behind the Scenes of Science. We're running most Tuesdays this fall. Next week, we'll be talking to Jeremy Miller, a scientist in the Allen Institute for Brain Science, who is helping develop a list, a taxonomy of all of the types of cells that are found in the brain. So Christina has just dropped the link in the chat where you can register on behalf of your class for the remaining sessions in this series. We're, again, running almost every Tuesday this fall. And thank you again, Mackenzie, for being here today. And I hope you have a great rest of your day. Thank you, everybody.
Bye.